Thank you everyone for carving a little bit of time from your busy schedule for today's presentation. As you can see, it is entitled Generating Tissue-Mimetic Environments to Accelerate the Production of Next Generation Biologics. Um, so today, the goal is for me to present some of the data that we have produced here at Ronawk and also of our collaborators throughout the world, actually. And hopefully this video is making you intrigued about what is that we're going to talk about. This is just one part of our product, which is the BioBlock. And you can see here, this is a three-dimensional recreation of images acquired with an AXR confocal. And you can see how the cells are interacting with each other, but also with our matrix forming this complex structures. And with that said, I would like to start introducing our product. What is a BioBlock? A BioBlock is a 3D printed hydrogel that has controllable properties that mimics the native tissue mechanics. It has four main characteristics: the macrostructure, which looks like a puzzle piece, and this facilitates the culture environment that we were able to develop. And this cultural environments eliminates the need for traditional subculturing, as well as reducing lab waste. And some examples of this is normally into the culture, tissue culture, you will need visual confirmation of confluency in order to pass the cells and, of course, expand that cell culture. In the case of culturing cells with our BioBlock, you will only have to add 1, 3, 4 BioBlocks depending on your culture vessel, and that way the cells migrate to the next empty area and start populating that area, expanding that way your culture without the need of adding trypsin or any agent that will release the cells from the culture vessel. By reducing lab waste, this is an excellent example of how we can reduce lab waste. You can fit up to 4 blocks in one well of a 6-well plate. That will be totaling 24 blocks in a 6-well plate. And that includes a maximum of 10 mils of media that you will be using, reducing the amount of plastic that you will be using for your culture, also the amount of media overall for an experiment. The second characteristic that I want to talk about is the microstructure. So the microstructure that we have developed is a network of microchannels that allows for transfer of cells, nutrients, and gases. And we're going to go a little bit deep on that on the next slide. But something I want to emphasize is that, as you can see here, that is one of our microchannels, and the line there is the hydrogel. Is that, as a difference to other 3D printed hydrogels, we don't embed the cells in the hydrogel. The cells actually coated the surroundings of the hydrogel, using our hydrogel just as a scaffold to form their own architectures and behave however they want to do it. Taking a little bit back of how the Bohr network allows for this mass transfer of nutrients, cells, and gas. These are some of the examples. In panel A, you can see that we have placed one of our BioBlocks in a tissue, and we added 1 mil of media, and we can see that within 2 seconds, that mil of media actually went through the entire block right there. And then on panel B, we can see that that transfer that was done with the media can be reflected with the cells, because when you see the interior of the block, you can see the cells surrounding the pores, around the inside the pores in the negative space. And once you get out of the inside of the block, you can see the cells also laying down on the matrix there, allowing for this very interesting behaviors and complex structure formation, not only in the outside, the exterior of the block, but also on the inside of the blocks. And in terms of gas exchange, we collaborated with O2M for this data here. And what we can see is that the diffusion of oxygen is very consistent throughout our block, meaning that your cells are getting the proper nutrients because of the fluid distribution, but also the proper gas exchange. This will allow for the cells to behave happily, healthy, and you will not be developing that necrotic core that characterize a lot of three-dimensional cultures. While the third and the fourth characteristic are the materials and the mechanics. These two characteristics specifically will work hand in hand, but when you combine them with our other two characteristics, the macrostructure and the microstructure, makes our product very unique. We offer three main materials. We can do a natural polymer, a synthetic polymer, and a combination, and each one of those will serve for a specific purpose. But most importantly, we can tailor its mechanics. What that means is that we can mimic the native tissue mechanics of the origin of your cells. That way, we allow for biochemicals and biomechanical cues that will enhance your cell culture experience. Our product was developed with the intention of being used with adipose stem cells. So right now, the elastic modulus of our product is around 3 kilopascals. And if you can see on the bottom panel right here, the importance of having those mechanotransductive signals is because the cells will behave differently depending on the mechanics that they are presented on. And especially when talking about stem cells or iPSCs, this will also affect how fast and how effectively they will differentiate into other cells that are of your interest. At this point, I'm hoping that you're coming to the conclusion that our goal was to develop a tissue mimicking environment that will facilitate and accelerate research. However, even though this was our main goal, this doesn't invalidate other forms of cultures. This includes 2D vessels. Without 2D culture, we wouldn't be here today. 100 years ago, when the first cell was cultured in a glass, it opens the possibility to discover more about cell biology and understand cell and development in general. However, as you may have encountered in your work or your graduate work, there are several challenges with 2D cultures. One of them is you have very issues trying to translate your research that you do there. There's only one mechanic, as I discussed before, with only having glass or plastic on it, and there's nothing in nature that is actually 2D. The cells are communicating within each other side by side, top to bottom. So it's not just one dimension there. Then you have other three-dimensional cultures like organoids and spheroids that over the past 10 years, they actually enhance our understanding of the need of three dimensionals, of the need of that sea, of the need of the cells talking to each other in many directions there. However, they're very hard to scale. They often don't propagate, and they have a mass transfer limitation at developing those necrotic cores. And now when you develop those type of cores, then you don't know if the data that you're getting out is due to what's going on the inside or what you actually were looking for. The other models that are available are animal models, and animal models are great. They have furthered our understanding of new drugs and the development of new diseases, but they are expensive, and of course, they involve a lot of ethical issues. And you might have encountered uh, people that do not agree with your use of animals in your research, even though they are very necessary. And our product, the BioBlock, stands right there in the middle of traditional cultures and animal models, giving you the sense of the easiness of culturing cells with the complexity of forming very different structures, the able to communicate with each other of a animal or three-dimensional model there. Now, the product is not entirely perfect, mainly because we are just finding out that 3D is necessary, and we don't have a lot of understanding on like how to analyze stuff that's going on in three dimensionals. So we are in desperate need of essays in order to assess not only our product but other three-dimensional culture models. And this is where a huge opportunity is for you guys that are present in this presentation, because you can help us develop those new essays. Now, the objective of this presentation, as I mentioned in the beginning, is for me to communicate the capabilities of Ronack's BioBlocks to unify research areas by facilitating collaborations for the advancement of new therapies, one puzzle piece at a time. As you may know, I'm a employee of Ronawk Incorporated, and also I just wanted to clarify and mention here that a lot of the data that I'm going to present is either in the process of being published, uh, or there's stuff that I cannot divulge with you guys. But we are willing to set aside some time and talk a little bit more one-on-one to discuss more of your needs of your project and how can we help you with it. Going back to unifying research areas, today my goal is to discuss how Ronawk has been impacting several research areas, specifically the ones mentioned here, and we're going to start with neuroscience research. So far, if you have been looking at our social media and all the things that we have been doing during the past couple of months and the beginning of the year, we have been working with several collaborators from different research areas, and specifically this here, where we were helping to develop a neuroblastoma tumor microenvironment. This collaborator here seeded both patient-derived neuroblastomas and primary Schwann cells in the BioBlock and cultured them for 4 weeks. The culture of the cells for 4 weeks, as you can see here, allow for the cells to start communicating with each other, but most importantly, interacting with each other. And this is a AXR confocal imaging of just one pore of one side of the block. And I emphasize a lot of that one pore, one side of the block. We're only imaging about 1% of the entirety of the block, which is 1 centimeter cube. And this is how much we're getting out of how the cells are interacting with each other. Other neuroscience research that we have been helping is with this collaborator. Is a biotech involved in gene therapies, and their goal was to expand the culture of primary neuronal cells. And these neuroprogenitor cells, you can see that within 7 days, we're starting to aggregate and form very complex structures. Sorry, by day 9, it's very impressive how they started forming this network-like patterns in the bioblock, filling in not only the insides of the channels, but also the outside exterior areas of the blocks. And by day 13, those networks were very complex and very full of cells, which allow for that very interesting behavior that we saw there. For other research areas that we have been working with, for cancer research, we're currently working with an academic collaborator based on Florida that is interested in developing a drug-resistant cell line for new therapeutics. So their hypothesis here, it was, since it takes up to a year to develop a resistant cell line in 2D, maybe if we add that three-dimensionality to it, would allow for the cells to accelerate that process of becoming resistant. And what they have seen so far is, in terms of cell culture, a proof of concept, cells are getting into the blocks, even though they have been exposed to the drug for the entirety of the culture. And you can see that by day 7, those microchannels were full of cell because of how dark those areas are. And by day 8, they were getting out almost aggregates out of them. The way that we designed this experiment, it was, we had what we call a mother block. What that means is an initial seeding block that we add other blocks to it so the cells can be passed, quote unquote, passed to, so the cells can expand more. And for every time point that I'm going to present on our next slide, they were taking out a daughter block, which were the blocks that were getting cells passed to by the expansion of the cells to a new culture area. And what we saw here is that over the different time points, there was an increase on the GI50, which is the concentration at which a drug kills half of the population. And that is very important there to define that is half of the population there. And we saw an increase on that GI50, meaning that the cells are taking more and more of that drug in order to get half of its population terminated. And this is just a visual representation of the data I just presented, and we can see that over time, for the cells that were cultured in 2D, there is a decrease on that GI50 value, while for the cells cultured in the bioblocks, we see an increase on 30 days and then a plateau. Hopefully, we continue getting this data. We are doing some changes to our experimental design in order to achieve that goal of keep increasing that GI50 value. Our third research area that we have been working is with iPSCs research. Most of our iPSCs research work has been with the focus of expanding the culture. And interesting enough, we have, and I'm going to present here, 3 different collaborators that work with iPSCs with the same goal, expanding that culture. The first collaborator, it was a biotech that focuses on gene therapy, and they are an international organization. And what they saw was that even by day 1, by 24 hours post-seeding, the cells started to aggregate in different areas of the block. By day 2, these aggregates were huge. They were really big. And very visible, even this is a 4x magnification. And by 4 days, those aggregates started to being expelled from the bioblocks. Now, at that point when we saw this, we didn't know what was going on, to be honest. And we thought it was a very interesting behavior that the cells were having. But since we didn't have uh, more information about this, we didn't know if it was the bioblock that induced this behavior or if the cells just did this by themselves. So another collaborator, which was Biotech with Multiple Divisions, also an international organization, cultured the cells in the blocks. In this case, we were able to get images 7 days post, and the same behavior was observed, where the cells started to aggregate, and by day 4 to 7, those aggregates started to get expelled from the bioblock. Now, this sparked our curiosity, and we were like, we need to do more research on this. Why is this going on? These are two different collaborators. They're getting exactly the same data. Then something's going on. The bioblock is allowing for the cells to behave this way. Why is that the case? Can we repeat this? And fortunately enough, we got another collaborator, this time an academic from Florida, and we work more hand in hand with them, trying to understand why this behavior was happening. And what we saw, it was that within 1 day, like our first collaborator, aggregates started to form. And that aggregate right there is from the center pore of our bioblock. By day 4, again, more aggregates are formed, especially near those pores. By day 7, like before, aggregates are very, very big. And by day 10, they're even visible by the naked eye, not even the need of a microscope right there. At this point, those aggregates started to get expelled from the bioblock, and you can see them here by day 15, where they are floating around the well that was seeded with the block, with the cells in. And what they decided to do was to connect an empty block right here and see how the cells migrated and proliferated just to the new cultural environment, and then proceeded to the original block to get differentiated. But before they differentiated the block, they decided to test those aggregates that were coming out of the block. They wanted to know if the cells that were coming out of the block were retaining their stem markers. And what they saw was a twofold increase on the stem cell markers for the cells that were coming out of the bioblocks, those iPSCs aggregates. And this took us by surprise. We thought that maybe the cells were bad or something like that, but no, the cells were so happy they were getting out of the blocks, and they were still retaining those stem cell markers, which meant that we could use those cells for further investigation. Unfortunately, in the terms of the differentiation, there was some problems with the reagents used, and differentiation didn't happen. But we are currently working with this collaborator to take this further, keep checking those stem markers for longer periods of time, and also differentiate not only in the bioblock, but also the cells that are coming out of the bioblock. Another research area that we have been seeing a lot of interesting stuff is just cell biology. Um, if I didn't mention this before, I'm gonna mention it now. But the bioblocks allow for new biology to appear in front of you. So it is very interesting to see how the cells behave, how different cells behave, how immortalized cells um, react to such environment, and how can we keep those primary cells so young and happy. So some of the things that we have been testing is for that delay of senescence. And what I mean by that is making sure that the cells are not getting old and not working properly. This is some of the research that we have done in-house over the past couple years. And what we see is that we did a quantification of fluorescence for senescence, and we culture the cells for different periods of times. So we culture the cells both in a 2D environment, in this case, a 6-well plate, while also culturing the cells in our product, the BioBlock. And then after the cells reach into the a certain passage, in this case, we're going to be talking about passage 2, 6, and 10. When the 2D reach passage 2, 6, and 10, the cells from the bioblock were extracted and seeded to then be tested for senescence. Now, the 2D culture was passage every 4 to 5 days. And what we're talking in 3D is a passage equivalent. So at the same time that those cells in 2D reach that passage that we are interested on, those cells were taken out of the three-dimensional environment and put on a plate for being tested for different uh, assays. As we can see here, by passage 2, even in 2D, we start seeing cells that are positive for that senescent marker, which is, we use beta-gal as our senescent marker. By passage 6, we see that increase in that senescence with a minimal increase in senescence for the three-dimensional matrix here. And by passage 10, that is significantly increased while we don't see that much change on the bioblock right here. And this data was then visualized like this, and we can see that the non-senescent cells stay stable over time in the bioblock, while in the 2D class, you can see how they start decreasing because their senescence starts increasing over time. So the bioblocks, when we culture specifically adipose stem cells, significantly limits the promotion of senescence over time. And this is really important, especially when talking about stem cells and primary cells. Now, we did this with different culture environments. We culture the cells in the bioblock and saw a minimal increase in senescence over time, while for the 2D culture, it is a significant increase on senescence over time. But we also did culture cells in a matrigel, and again, we see that increase in senescence over time. And when we do them in spheroids, the same thing is visualized. And again, this was when the cells reach certain passages, we take them out of their corresponding matrix or culture vessel and then test for senescence. Another thing is the retainment of phenotypes when we're talking about cell behavior. We want to make sure that every single time that we're passing the cells or for how long the cells are cultured in the bioblock, you still are retaining those phenotypes that are very important for you to perform your experiments. In this case, we were looking for stem-like phenotypes. All of this data that I'm showing here has been done both with fluorescent markers and PCR, and you can find them at the end of the presentation. I will have all the listed references for them. And what we can see here is that even a passage 10, which is a 4-week, passage 10 is a 6-week culture, you can see that the cells are retaining those markers right there, not only for the CD73, but also for CD90 and CD105, while the 2D culture starts very strong and then starts losing that marker right there. We took it even further, especially with ASCs, which are a type of a mesenchymal stem cell, and we wanted to know if the cells, while being cultured in these different vessels and culture environments, if they were lose their potential to differentiate. And what we see here is before we proceeded with differentiation protocol, we saw that there's a retainment on that potential for the cells culture in the bioblocks to differentiate into the different lineages. And that was then confirmed by the differentiation protocol that is shown here, where you can see that for all the lineages, the osteogenic, the adipogenic, and the chondrogenic, the bioblock is the most consistent and higher population of cells for the different differentiation, while the other culture environments have a more lacking preservation of that potential for the cells to differentiate there. Now, another research area that we're interested on, especially in-house, is the wound healing. And for this, we focus on the use of mesenchymal stem cells and their potential secretome to help with wound healing new therapies. And what we're going to focus here is on that secretome. And what we saw is that while seeding cells in the bioblock, you have a 15% increase in soluble protein secretion, a 25% increase in the secretion of antioxidants, and a 4 to 5 increase of secretion of extracellular vesicles right there. So one of the experiments that we did was we culture adipose-derived mesenchymal stem cells in both our product, the bioblock, and a 2D flask. And every week, media was collected from both. And that media that was collected was then used to treat keratinocytes that were cultured in a 2D environment. And something that we saw there was an increase in the metabolic activity for the cells, for the keratinocytes that were treated with the media that came from the bioblock, an increase in proliferation for the cells, for the keratinocytes that were treated with the media of the bioblock and an increase in the migration for those cells as well. It was almost 100% increase on metabolic, 15% increase in proliferation, and a 45% increase in migratory. And if you're interested in working with us and understanding more of your cells, how your cells behave, and just curious of how much you can do, there's new research possibilities that you can help us with to add more to our story here. With that said, the takeaway message, coming back to my objective of communicating with you guys the capability of Ronack's BioBlocks to unify a research area by facilitating the collaborations for the advancements of new therapies, one puzzle piece at a time. Some of the things that we discussed in this presentation were that we were able to develop a tissue-mimetic environment that preserves cell phenotype, that delays cell senescence, that increases the production of biologics, that encourages in vivo cell behaviors, and that it allows for cells to thrive in harsh environments. And all of this is possible because the cells are being cultured in a more native environment. With that said, I would like to thank the Ronawk team. Without them, I couldn't be here and we wouldn't have all this data to show. So thank you so much. And if there is any questions or you want to have a more one-on-one talk with us, please contact Scott slay@ronawk.com, and here are the publications that I talked about. They go more in depth on how the bioblock helps with the culture of cells, the production of biologics, and the retainment of phenotypes and the delay of senescence. And thank you so much.
Thank you, Aidyn. Can you hear me?
Yeah.
Great. We have a list of questions, and I'm going to read them off for you one at a time, and let's see if we can answer as many as possible in the time we have remaining. To all the participants, if you want to drop questions in the chat, that would be great. You'll find the chat at the top of the banner. So let's get started. Aidyn, what evidence do you have that shows why bioblocks are even more in vivo-like than organoids and spheroids?
Oh, that's an excellent question. At this point, what we're noticing is, especially doing some histology on sections of the bioblocks and also on cells that have been expelled from the bioblock, is that they, histological, they look a lot like tissue. Like the tissue you will get from an animal model or even a human sample. So that is so far our evidence. We keep getting and collecting more of that evidence, looking more for the functionality of that tissue. And that is our current main goal, trying to prove that the tissue that it's being formed is functional, so it adheres to the definition of tissue.
Great, thank you. Second question, Aidyn, is how did you passage 3D cells?
That's an excellent question. In the case of the bioblock, you will just add another bioblock to it. Because of the puzzle piece shape, they will connect, and that way the cells will migrate from one bioblock from the mother bioblock to the daughter bioblock to populate that daughter bioblock. Now, what we recommend is that you just not add one, add several. That way you can use those daughter blocks for the experiments that you need. But this will retain and maintain a constant culture of your cells.
So, Aidyn, I'd like to clarify two things on that. So this means you don't trypsinize the cells to do a passage, correct?
Correct.
And then the second is something you didn't mention is how long have we been able to keep cultures going by just adding and subtracting blocks?
Oh, yes, we have done a continuous culture for up to 6 months.
Thank you. Next question. Do you think that delayed senescence is because of the factors in the hydrogel or because of the 3D culture?
It's a combination. So as I mentioned, uh, one of the main characteristics of our product is having the material. The material is sending biochemical cues for the cells to understand that they are in a environment that is safe, and then the mechanics is sending their cues saying that it is a three-dimensional environment. So your delay in senescence, it is due to both things, the composition of the bioblock, but also the three-dimensionality of the bioblock.
So Aidyn, I love this next question because it's about staining, but I really think it's about how um, we can extrapolate that to how anything enters and moves throughout the block. The question is, how did you do the staining for the differentiation experiment on the cells cultured in the bioblock?
For that particular experiment, cells were extracted from the bioblock, and the staining was done uh, in a plate. That was the decision to do that. It was so we treated all of our cells the same way, because the cells that were cultured in the matrigel needed to get extracted from the matrigel, and the cells cultured in spheroids needed to get dissociated. So we also decided to take out the cells of the bioblock. With that said, I know I mentioned at the beginning that we offer three different materials. Our natural polymer, it is degradable, so we're able to extract the cells out of the bioblock, because we have also developed a extraction reagent that only targets the matrix and not the cells, allowing for us to culture, to use those cells for other things, especially when comparing different culture environments. Now, I want to add more to this. Uh, Staining cells in the bioblock is as easy as if you were doing a whole in situ staining of your tissue. So if you have stained a good example, brain, you can do that with the bioblock also. We just recommend that your staining is a little bit longer than that you will do for 2D cell culture, and we have been working over the past couple of years developing those protocols, and we are happy to share them with you.
Great. Next question, how did you perform gene expression analysis?
Again, because we're able to extract the cells of the bioblock, that's how we did the gene expression.
How did you test the metabolic?
That is an excellent question that I will defer to my collaborator, Dr. Hutch, that is not here. So if you want more information about that, feel free to reach out back to us so we can get you that answer.
I would imagine that's also in the publications that we have, which we'll share at any time, and we'll share after this to all participations how to read or how to achieve, I'm sorry, how to access those publications. Next question is regarding drug-resistant experiments. An increase in GI50 is generally associated with de novo mutations. Were the cells sequenced to identify any underlying mutations? Also, have you tested if cells grown in bioblocks are genetically more stable than traditional 2D?
Oh, that's an excellent question. So to be honest, to our understanding and what it was shared with us, no, they didn't test for any genetic mutations. However, I appreciate you pointing out that, so we can talk about that with our collaborators and see if we can see any changes there. So thank you so much for your input there. In regards to making our making the cells in the bioblock more genetically stable, that is an excellent question that I don't have an answer for, but we are currently working with other collaborators to see different changes in their genetic composition of the cells being cultured in the bioblocks.
Great. Next question. Could this system be set up for high throughput assays? Meaning, could you scale to perform drug screening on, say, 100,000 compounds? And what would be the cost compared with similar organoid and spheroid models? Aidyn, why don't you take the first two questions and I can answer that third one.
Yes. So the sky is the limit. If you're willing to work with us to develop that high throughput assay, we're here for you. So give us a call.
Very true. Now, to answer the cost comparison, we have done some models on cost comparison. We reduced the media consumption and consumable consumption 80 to 90% to develop cells. So the overall cost, we assume in a drug assay of that size would be about 1/10 the cost compared to scaling using roller bottles, bioreactors, etc. We haven't gotten there, but when we're comparing small scale, that's proving out to be true. The other advantage in scaling is you don't change the substrate. You stay in that same substrate from discovery through scaling and trials. So great question. Next question is, and I think it's really the same one, what is the outlook for large scale applications? I'm going to answer that one, Aidyn. We can do things in relative, for an academic lab, it would be large scale, but for industry, we're small scale right now. We do have models and designs to scale and create flow through our blocks. They have been built, they've been tested, we have not found the application yet. So it's a great question of large scale applications. They're there. We believe we would reduce the space, the labor, and the material significantly in that application. We just have to go from where we're at now to get to there. So it's a great question. Aidyn, next question is, how is efficiency to release the cells from bioblocks?
Can you repeat the question again? Sorry.
How is the efficiency to release the cells from bioblocks?
That is an excellent question, especially because I think it was the first question, and how are we making sure that what we're having is tissue-like. So interesting enough, the longer the cells are cultured in the bioblocks, the more difficult they are to get out of the bioblock. The reason being is the older the culture gets, the more complex that tissue-like structure is, and you have to treat it like tissue at that point. So you will have to implement tissue dissociation techniques in order to get cells out of there. The younger the culture is, the easier it is. It takes around 2 hours to dissociate completely the block and get out the cells. Normally, the viability of those cells is retained in between 98 to 99%.
Great. How thick are the bioblocks, and is the oxygen tension the same throughout the depth of the bioblocks?
Oh, you missed our couple of first slides. Yes, so our bioblock is 1 centimeter cube. And yes, the oxygen is retained throughout the entirety of the bioblock. We did a study with O2M where they tested the oxygen diffusion throughout the block, and it's pretty consistent.
Just to this point too, I know some people jumped on halfway. I am going to be putting this presentation on YouTube as fast as possible, and we will be sharing the complete recording with everybody as fast as we possibly can get it out. So if you missed something in the beginning, you'll have access to everything. Next question, senescence. Have you looked at the possibilities of stress-induced premature senescence in your iPSCs when you grow in your bioblocks?
Thank you. As I mentioned, we haven't done the iPSC work in-house. So that will be something to relate to our collaborators and see if we can test for it. So thank you for the suggestion. I'm gonna talk it forward.
Great. And we got 2 more questions. Everyone, if you didn't get a question or you have one, if you go to the top of the chat, you'll find my email as well as a way to schedule a meeting with us. We'd love to talk to you. We want to share our technology and give it out as quickly as we possibly can. So last question is, if you do last 2 questions, if you do 3D staining, will imaging be an issue? I meant the transparent might be an issue.
So our bioblock, it is pretty transparent. It has a refractive index of 1.3, if I'm not wrong. So imaging in the bioblock is quite easy. There are some limitations, and the reason for the limitations is how big our bioblock is. As I mentioned, it is 1 centimeter cube. So penetrating light throughout the entire bioblock, it is very difficult. Nevertheless, we're working in partnerships with, in this case, Nikon, to help improve this problem. And so far, we have gone to about almost 5% in deep of the bioblock. And we continue to improve this with more days that we are practicing our imaging techniques with the bioblock.
Thank you very much. We really enjoyed this. As I mentioned at the beginning of this webinar, our goal is to break this down into some smaller bites. Let's focus just on exosomes. Let's focus just on iPSCs as we get more data. Please connect with us through my email or through setting up meetings. Follow us on LinkedIn. We think we really have something exciting to share, and we want to put this tool in as many hands as possible. So thank you very much, Dr. Medina-Lopez, for presenting, and thank you for all the participants.